Hello everyone, and today we're talking about a latest paper published in Science. The title of this article is Contrastive Machine Learning Reveals the Structure of Neuroanatomical Variation Within Autism. Let's jump straight into the article. Before discussing the model, let us look at the motivation behind all this. We know that in medical field, sometimes it is hard for computers to diagnose because they often got confused by variations among the vast human population. For example, think about any sort of skin lesion. Without a priori knowledge that humans have different skin tones, machine learning models might get confused. Similarly, when trying to understand autism spectrum disorder, typical machine learning models suffer because when they look at the MRI scans, they might mislearn the features that are common to the general population, which gets in the way of the less obvious features that are specific to ASD subjects. So, where's the motivation to encourage the model to learn to tell the difference between what features are common to the general population and what features are only specific to ASD subjects? The authors choose to use CVAE. As you can see here, I already have the architecture laid out here. It is a variational autoencoder, aka VAE. But what is different here is the C, which stands for contrastive. And by the end of this video, I hope you can understand what the contrastive here means. So let's take a look at this graph here. We have the encoder, the latent features, and the decoder. A very typical encoder-decoder structure, right? But this very model here is a little bit different. As you might notice here, it has two segregated encoders. One encoder maps the input to latent features that the model thinks are common to the general population, in this case common to both the typical control and ASD subjects. And the other encoder maps the typical control, maps the input to the latent features that the model thinks are specific to only ASD subjects. To train the model, the author prepared a dataset that consists of both ASD subjects and typical controls. Typical control samples go through only the encoder for shared features, while the ASD subjects go through both encoders. If you find it hard to justify why the data path should look like this, then think of typical controls which the model learned the shared features from as the background of a picture, and ASD subjects which the model learns both the shared feature and the ASD specific features from as a picture with both the foreground and the background. Similar to other VAEs, contrastive VAE is also probabilistic. In other words, instead of producing a definite latent vector, the encoder actually yields a distribution for the latent vector. In order to do backprop on this model, the reparameterization trick is used, where a sample is obtained from this distribution and then passed on to the decoder during forward pass. The decoder then decodes the feature and tries to reconstruct the input. The reconstruction loss used here is the MSE loss. After finished training the model, the author attempts to make sense of the latent features. And first and foremost, they want to validate these features. Are the entangled features really disentangled? Does the latent features learned by the model make clinical sense? To answer this question, the authors use what is called representational similarity analysis, aka RSA. Before introducing what RSA is, I would like to first introduce the idea of representational dissimilarity matrix, aka RDM. Let us say we have a feature, a group of subjects, and the definition of similarity or dissimilarities for this feature. We can calculate the dissimilarities based on this feature between each pair of subjects. Let's say we have n subjects. Then we will have a matrix of size n by n that characterizes the variation captured by this feature within the group of subjects. With this concept in mind, let us move on to see how the authors are using RDMs. Remember that VAEs are probabilistic, which means the encoder, instead of producing a definite latent vectors, actually yields a distribution of latent vectors which we can sample from. The authors then decide to sample 10 times from this distribution for each subject and calculate an RDM every time they sample. Also reminded that we have a shared latent features and specific latent feature. So they actually calculate 2 RDMs every time they sample. One RDM is based on shared features and the other is based on specific features. Now you might want to pause here and ask, what does an RDM mean after all? Why are we calculating these matrices? To answer this question, I will say an RDM based on a specific feature shows the variation of the subjects captured by this feature. This concept is very useful later on. Now we would like to know if the shared and specific features learned by the models are actually legit in a clinical sense. To do this, we're essentially asking this question: how similar is the variation captured by the learned features compared to the variation captured by the properties of a patient, such as gender, age, symptoms, and etc. Reminded that variation captured by a feature is essentially the RDM based on that features. So we're trying to measure the similarities between 2 RDMs. To quantify the similarity here, the author used Kendall's coefficient, tau. There's a lot of stuff going on here. To clear things up a bit, I will just use an example from the paper to illustrate what the author are trying to do here. So in this specific experiment here, the authors are trying to compare the variation among the subjects based on the ADOS total score to that based on shared features and to that based on specific features. So it seems necessary before we move on to the next step to obtain an RDM based on ADOS total. Now let's pause and take a look at what we have at hand and what questions we want to answer. We have 10 RDMs based on shared latent features, 10 RDMs based on specific latent features, 1 RDM based on ADOS total, and a tool called Kendall's coefficient, which can be used to calculate the similarity between 2 RDMs. And the question being asked here are, firstly, is the variation captured by the shared latent features similar to the variation captured by the ADOS total? In other words, is the similarity significant? And secondly, is the variation captured by specific latent features similar to the variation captured by the ADOS total? Again, in other words, is the similarity significant? So let us dig in. We first calculate 10 taus between the 10 RDMs based on shared latent features and the RDM based on ADOS total. Now we have 10 taus. And to answer the question of significance, we have our good friend t-test. Specifically, in this case, a one-sample t-test. And in this case, the null hypothesis is that Kendall's coefficient tau equals 0. From now on, all we have to do is simple statistics. We calculate the t-statistics and the corresponding p-value. If the p-value is less than the significance level, which the author sets to 0.001, we reject the null hypothesis and claim that the similarity is significant. And we repeat the same process for the 10 RDMs based on specific latent features too. And we can answer the question of whether the similarity between the variation captured by the specific features and the variation captured by ADOS total is significant. Now let's take a look at this figure here. The authors report that for shared features, the similarity is not significant, whereas for ASD specific features, this similarity is significant. Since ADOS total score is a measure of ASD clinical symptoms and the model's ASD-specific features are associated with ADOS total score, we can give a model a pat on the back. Now the authors want to take a step forward and ask, is the difference between the two, or we should probably say 20, similarities or taus calculated before statistically significant? To answer this question, a paired sample t-test is needed. Reminded that we have two groups of 10 taus. Now what we need to do is just calculating the t-statistics and the corresponding p-value. Again, let us take a look at the figure here. We see that the difference between the shared and specific latent features is significant, as is signified by the triple stars. Now we understand how RSA works. Let us try to interpret these results and also trying to decide whether we should really give the CVAE model a pat on the back. First, we have here scanner type as the property. Intuitively, scanner type is a property that is common to both TCs and ASD subjects. Therefore, it should be more associated with the shared latent features. And as we can see in the picture, there is really a significance indicated by the 3 stars. And it is indeed more similar to the shared latent features compared to specific latent features. Next, the author move on to look at the measures of clinical symptoms. We have already looked at one example above, which is the ADOS total. There are 2 more measures in this category mentioned by the authors, which are DSM-4 subtypes and Vineland questionnaire. Analysis of these 2 cases is not hard and is left to the readers as homework assignments. The authors actually conducted a very in-depth RSA of various neuroanatomical features and patients' properties, and what I mentioned here are only 2 examples. But from these 2 examples, we can already start to appreciate the model's ability to tell ASD-specific features apart from features that are common to the general population. I would give this model a pat on the back. In the following videos, I might take a closer look at the CVAE model architecture or do a similar video on the rest of the paper. But as for now, I would like to conclude and thank you for watching.